Hello, my name is Shelby Harris, and today I'll be talking to you about CAR T therapy. CAR T therapy is a form of immunotherapy that uses altered T cells with special CAR receptors on their surface. CAR stands for chimeric antibody receptors. They are made in the lab specifically to target the disease at hand for the patient. How it works is that T cells are collected from the patient's blood, and then they are fused with the CARs specific to the disease to be expressed on the cell surface via gene editing. The new CAR T cells will then be transfused back into the patient, which would allow the CAR T cells to encounter the cancer as if they were natural T cells in the body. CAR T cell therapy is FDA approved for treatments of non aggressive Hodgkin lymphomas, yes, CARTA, and leukemia that has been previously failed treatments in younger patients than 25 with Chimera. Overview of T cells. So, what T cells are is they are white blood cells that are trained in the thymus to respond to self and non self antigens. There's two main types, which is the CD4, which is the helper T cells, which recognizes antigens presented on MHC class 2 molecules, and CD8s, which are the cytotoxic T cells, which recognize antigens on MHC class 1 molecules. So helper T cells recognize other T cells, your B cells and stuff like that, professional antigen presenting cells, versus cytotoxic T cells recognize every cell in the body because every cell in the body has class 1 versus not every cell in the body has class 2. Cytotoxic T cells attack cancer because cancer is MHC class 1 presenting self cells. However, tumor cells have developed a number of ways to avoid being found by the immune system, which they include MHC modifications. So one thing that they'll do is that they'll deregulate MHC class 1 expression, and or they'll upregulate it. So they'll either have no MHC class molecules on there, or they'll have so many that the T cell can possibly not bind to the cell. And another way they do it is they release immunorepressive products, so they release cytokines that calm down the T cells. The T cells don't recognize that they're cancer cells. So, chimeric antigen receptors are lab made antibody receptors that are specially designed to target cancer tumor cells. So, chimeric means that they will have both antigen binding cells and T cell activating functions all in one receptor, versus in the body, there has to be two separate receptors in your natural T cells. CAR T cells can be from the patient's own T cells, which are autologous T cells, or from a healthy donor, allogenic. So, and usually when you're doing CAR T therapy, you take the patient's own T cells so you don't get rejection. But you can take them from a healthy donor and have like them pre-made to be transfused into the patient already, so that the patient might have a chance to start the treatment before they get their own made. CARs have the four main regions of their receptor, which is the antigen receptor, the hinge region, the transmembrane domain, and an intracellular T cell signaling domain, all in their receptor. So here we have a CAR, and the top of the CAR right here is the antigen receptor. This binds to the antigen and begins their immune response using the antibody variable heavy chains and light chains. Here we have what's called the single chain fragment variable, or the SCFV. So this is essentially the antibody of the CAR cell. Does the same thing as what an antibody would do on a natural cell. The hinge, or the spacer right here, is very important for the flexibility of the CAR. So instead of being, if it has a long spacer, instead of being rigid to the cell, it can move around and bind better to things. The transmembrane domain is the bridge in between the receptor region and the interplasmic region. So the transmembrane is not only anchors it to the cell, but also does the signals across the cell membrane into the cell to further the response. And then the intracytoplasmic signaling domain has the molecules necessary for T cell activation, like the ITAMs and the CD3 zeta, and other co-stimulatory molecules like CD28 will all be in here in the cell to get stimulated when something binds to the top part and to activate the T cell itself. Here's just a generation of cars, just to see how they've grown over the years. They've gotten larger because we've added more co-stimulatory molecules to them to better their activation functions and stuff like that. The top really has stayed the same. It's really a good design, but we've added more down here to better the response. And I also want you to notice that the fourth generations now are called trucks. And I'll tell you later what trucks means, but they're now called trucks instead of cars. So how does the therapy work? So to begin, the blood is drawn from the patient or the donor, and the T cells are isolated then from the blood. Then T cells are sent to a lab where they're transfected with genes that encode for the CARs and using an inactivated virus. And then when those cells are transfected, they're allowed to transform with that new genetic information to allow them to present those receptors on their cell membrane surface. So, and then after they've transfected, transformed, and they have started to show those molecules, they're allowed to grow up in culture in the lab, and they grow them up in numbers till you get enough numbers to transfuse back into the patient. So patients are treated with low-dose chemotherapy so that there's nothing else in the body other than the cancer that the immune system would have to deal with. And then the transformed T cells are transfused back into the patient's blood, where the CAR T cells can then act as a regular T cell in the body. This process takes about 3 to 4 weeks from isolating the white blood cells from the blood to getting them transfused back into the patient. So here's just a picture of it. So to start off over here, blood is taken from the patient and the T cells are isolated and sent to the lab. Using a virus, you transfect new genetic information into the T cell, which then the T cell does, the translation of the proteins into there, and then they show up on the outside of the surface as the new CAR receptor. They're grown up in numbers, they're allowed to populate in lab, and then they're transfused back into the patient to where in the body they'll encounter their cancer cells and kill it, like a normal T cell would. So current treatments that are now using CAR T cells are Yescarta, which was the first CAR T therapy for adults with non-Hodgkin's lymphoma that have failed at least 2 other types of treatment. So they've already gone through treatment and failed, that their cancer was not going away. 72% of the 101 patients in the trial entered some sort of type of remission after the treatment, with 51% of that being complete remission of their cancer. So 51% of the people who entered remission of the 101 went into complete remission of their cancer. Patients responded to treatment within a month, so it was actually very, very short timing, and had ongoing protection from the cancer due to memory cells being formed. So like in the regular body, when you encounter, an infection or something like that, you have memory cells that are being formed in your body. When you encounter that again, you have a better response to that now because you have those memory cells there to instantly attack it. So it was found that up into 2 years, it was a long time, that the memory cells could be found inside the patient's body. And then this is their logo I included, which is just a T cell with the CAR, and then we have a CAR T cell being made right here. That's their logo. Another treatment is Humira. So Humira is approved for children and young adults with B-cell leukemia cancer that have also failed previous treatment. So they also went through treatment, and also their cancer did not go into remission. So very aggressive cancer. The T cells were made to recognize the CD19 on the B cells in their leukemia. And 83%, which was 52 of the 63 patients that went into remission, had ongoing protection years after they received the therapy. 100% of those children and young adults in remission after Humira had undetectable levels of cancer left in their bone marrow, meaning that they were completely in remission. 98%, which was only one child out of the 65 who entered remission, received this MIDR negative status. So only one child went back into the cancer after they were in remission. And then I also include their logo. Since this is made for children and stuff like that, they have a superhero logo, which is very, very childish. And it has a good point that Chimera has helped kids with the same type of cancer that you have. But not every experience is the same, and many kids, it has helped them beat their cancer. So we do have to keep in mind that not every single person's cancer is exactly the same. So, a deadly side effect of the CAR T cells is what's known as cytokine release syndrome, or CRS. And essentially, what happens when your T cells, are going through immune response, they release cytokines, is what they do. And patients that are already weak from fighting cancer can be easily killed by this. So if you've been battling cancer for 2 years, you've gone through chemotherapy and it's failed, and now you're going through this new treatment, and when you get it, you have this cytokine storm from these T cells being formed, and you're already weak from fighting this cancer. It could easily do you in, and like that. So that was one thing that they really looked at, is to control the side effect. So CRS is being treated with ligand 6 that inhibits the master release of cytokines that can harm the patient. So if we release them interleukin 6, that they will have a lesser cytokine storm response, and they would have a better chance of surviving. Control of cytokine release syndrome is very important, as it may harm or even kill the patients. Like I said, if you're already weak from fighting cancer, you don't want this to happen to you. So the future of CAR T. So optimization of CAR T therapy is the main focus of the research. So to better it and make it faster and more accurate is what really researchers want. A CRISPR-Cas9 system is being developed for faster and better transformation of cells. CRISPR-Cas9 is very efficient and very accurate in transforming cells. It's a very good system to use. Dual therapies, in which CAR T cells now have 2 receptors instead of just 1 receptor on them, so they can better have a higher chance of responding to things by having 2 receptors instead of just 1. Decreasing the time needed for CAR T therapy would benefit the patients because, of course, like I said earlier, if you've already been fighting cancer for, say, a year and you're already weak, you might not have a month to wait for your CAR T to begin. You might not have that time to wait for the cells to be grown up in lab. You could die while they're in lab being grown. So if we can decrease the time needed to grow up the cells and make them, then we can increase the likelihood of our patients not passing away before we get those cells to them. Melanoma treatments are now being studied, because really right now it's just been Hodgkin's lymphoma and leukemia that has really been studied in this. Like I said earlier, trucks have now been formed, which is known as T cells redirecting for universal cytokine killing in armored cars. So trucks, what they have done now is that they've put in cytokine killing mechanisms already into the CAR receptor, so that way they don't have to control the cytokine release syndrome. It happens naturally by itself via the car itself, via the truck. And the armored cars are used for solid tumor attacks by being really fortified so they can break into tumors and stuff like that through thick tumors. And I believe that CAR T therapy has an extremely bright future in individual treatment of cancer. So, like I said earlier, everybody's cancer is different. When you treat cancer with a cookie cutter chemotherapy and stuff like that, you have a higher chance of it not responding to your cancer, because cancer is so individual to the person. With CAR T, since this is your own T cells using an own receptor to your own cancer, it's making it very individualized, and it's bettering the cancer treatment to the patient. And I feel like that would be an extremely good thing, and it would be something that researchers continue to research in, and I think it has a really extremely bright future in research. And here are my sources. So, alright, thank you for watching my video. I hope you enjoyed it.